All right.
Yes,
I
we
can
can
Hi.
see it.
Yep. Okay, great. Hi, my name is Duncan Brown. I am working on the NIFWIF Headwaters project that Marina mentioned. So I'll be talking about how we're evaluating headwater biodiversity and resilience to inform conservation in the Delaware Basin. So just to briefly introduce myself, my name is Duncan Brown. Again, I am a macroinvertebrate taxonomist here at the Academy of Natural Sciences. I specialize in a group called Chironomidae. This is an insect family. I have a few pictures down here at the bottom so you can see. Some of these are commonly referred to as bloodworms, which people might be familiar with, especially if you keep fish. They're called bloodworms because they have hemoglobin or something similar to hemoglobin in their blood just like we do, and that's what gives it that red color. And it allows them to survive in very low oxygen environments. And I'll talk a bit more about chironomids later, as they are very important for uh, macroinvertebrate communities. And as far as education, I have my master's in entomology from the University of Delaware and a bachelor's in ecology from Kutztown University. So the purpose of this project is to help us better understand the biodiversity of headwater streams in the upper Delaware. There's a lot still we don't know about the organisms that live in these streams, and these streams are especially important because this is like the source water for larger streams and ultimately the Delaware River. So things that happen here have downstream effects later on, ultimately in the actual Delaware. Another purpose is to determine how temperature changes, especially temperature increases, affect these freshwater communities. We expect that in the next few years, we're going to see temperatures increasing, and we really want to know what kind of effects that's going to have. And then we also want to collaborate with scientists and community partners to help improve conservation practices so that we can try to maintain healthy, uh, especially healthy streams, but just ecosystems in general. So for this project, we currently have 45 total sites, although we do have a few sites not currently on this list that were sampled back in 2001 and 2002. But we have 45 now, half of which you can see here on this map. These are all the northernmost sites, but I want to focus on four particular sites for this presentation. Just figure it'd be easiest to have a few representative ones. So that will be Cold Spring Creek. That is our one of our northernmost sites. The Upper East Branch of Callicoon, Fuller Creek, and Abe Wood. When this project was being designed back in probably 2020, this was before I started, they set some parameters for how they were going to select sites because we want to monitor healthy headwater sites because that helps us understand as things change, as temperatures increase, what changes we see, we can hopefully attribute to fewer factors. So, one important thing is all the sites are in areas where there is at least 50% forest cover. There is relatively little wetland cover. And one of the most important things is that there are zero major point sources upstream of our sites. That just means that there are no major sources of pollution getting into the streams. Obviously, that would have a big impact. And the more variables, the more factors, the harder it is to really suss out which specific variables could be causing the changes to macroinvertebrate communities and the stream health overall. So this is something they found when they first went out and sampled back to 2021 and 2022. They found that the more disturbed land there is above a site, the higher the temperature variability. And that makes sense. Disturbed land often has less plant cover. Less plant cover means less shade. That means that as the day gets hotter, the water gets hotter. Whereas in more shaded areas, areas in more forested land, they have a lot of canopy cover. That means a lot of shade over the water, which means that the water doesn't increase in temperature as much as it does when there's just direct sunlight. And so we wanted to limit this kind of variability as much as possible so that we know that the actual temperature variable that we're looking at is more like climate temperature changes and not so much day to day from day to night changes. So macroinvertebrates, that is what I specifically work with. These are invertebrates large enough to be seen with the naked eye, and in freshwater streams, that is largely going to be insects, crayfish, worms, snails, and bivalves, so freshwater mussels and clams. But by far the most common macroinvertebrates you will find in all of these headwater streams are insects. Uh, Typically, it's going to be 90% or more insect specimens. We don't see a lot of bivalves. There are, I would say most sites, I don't see any bivalves at all. We see a few snails. We do see a lot of worms and occasional crayfish, but by far it is mostly the insects. So that's things like dragonflies, which you can see here in this first image on the left. These are two dragonfly nymphs, and then a crayfish in the middle, and then this is a stonefly nymph on the far right. And an important term for using macroinvertebrates to determine stream health is the EPT taxa. You'll hear me talk about it in a little bit. That just stands for Ephemeroptera, which are the mayflies, Plecoptera, which are the stoneflies, and Trichoptera, which are the caddisflies. And these three orders of insects are especially important because uh, they are often very sensitive to environmental changes and pollution. So their presence tells us a bit about what kind of pollution is present, if any. And they are also often very numerous in our sites. So the more we have of them, the better our data is. I just want to talk about some of the more common taxa I have tended to find in our samples. One, this is very common, is the genus Baetis. These are the blue-winged olive mayflies. Very, very common. Here you can see on the left, this is the nymph stage. So when they're actually living in the water, this is what they look like. Typically pretty easy to identify mayfly nymphs because their abdomen has gills, and their abdomen typically ends in three tails, although sometimes two. And then on the right, we have the adult form of the mayfly. Another fairly common one, although not nearly as common as the mayflies, is the genus Nigronia. These are the dark fish flies. If anyone here is familiar with Dobson flies, these are very similar, very closely related, but a bit smaller. The larvae, you can see here, again, aquatic, these are all aquatic, are called helgramites and often can be very, very large. This is the genus Rhyocophila. These are the green sedges. This is a caddisfly. If you're familiar with caddisflies, a lot of them will build cases for their larvae, and they use that for protection. But this particular genus, Rhyocophila, are actually free living. So you can see the larva here has no case. They actually don't build cases. They just live like this before eventually pupating and emerging as the adult caddisfly. And by far the most common taxa you will find in any of our samples is the family Chironomidae. I had mentioned them earlier. This is the group I specialize in. They're the non-biting midges. The adults are pretty small. They often look a lot like mosquitoes, but as their common name implies, they don't bite. And the larvae are extremely numerous. It is not uncommon for us to have a sample of, say, like 200 specimens from one site, and 100 of them could be chironomid larvae. And they tend to be pretty resilient to environmental changes. They're very good at surviving with low oxygen levels, and they can tolerate pollution pretty well. And here, this graph shows just how much greater the chironomid presence is than all of the other groups. So you can see here that chironomids, these I believe is from the 2021 and 2022 collections, so not from our data last year, but I expect that data will look very similar. We have more than twice the number of chironomids as we have of the next most numerous taxa. I want to talk a bit about our methods of how we actually collect our data. First, we get to the site, and you can see in this top picture here that white tape. That is a 100-meter tape that we run down the length of the stream. That 100 meters is our transect, so we work entirely within that space. We collect some habitat data, so information about nearby human development, whether there are houses, farmland, things like that. We have to determine the substrate type at 11 spots along that transect. Uh, so whether the bottom of the stream is largely gravel, sand, mud, if there are plants present, that kind of thing, because that's very important for what we expect to see in our macroinvertebrate samples. And then also stream depth and width at those same 11 spots along the transect. For water parameters, we use a device called the YSI to get things like temperature, pH, and dissolved oxygen. Temperature, of course, is particularly important. We also collected eDNA, that's just short for environmental DNA, where we just go into the stream, we take a water sample, we filter roughly 3 liters through a small filter. And as we filter it, organic material, debris, bits of anything floating in the water gets collected on the filter, and that can be analyzed for DNA. And that lets us know a very wide variety of organisms that are present in the environment that we could never really determine just by going in and hand collecting macroinvertebrates. So it gives us a much wider view. And another nice thing about it is it also gives us some information about some terrestrial species because terrestrial DNA can still end up in the water. Either the organisms fall in and die and start to decompose, or something could eat them and then defecate in the water, and then you have DNA that way. For the actual macroinvertebrate specimens we collect, we have 2 different methods. One is called a Serber sampler. So if you see in this first image on the left, this little metal square with a net attached to it, that helps us get a semi-quantitative sample. So we can actually get some estimates on the density of the organisms present because we only collect from in this square here. So we know exactly how much area we have collected the specimens from, and that gives us an idea of how dense they are. But we also use a method called multi-habitat, where we can't really get any information about density because it's not quantitative at all. We just go in with a di D-frame kick net and collect from as many different habitats within the transect as we can find. And that way we get a hopefully the widest variety of organisms we can, because if you're just going in with the server, you're probably going to miss some things, especially when you go to areas that have, say, plants in some spots or have undercut banks where you can't really use the server. There are going to be organisms that live in that area that you just will never find without using multiple methods. And then we take the specimens back to the lab, we sift through, make sure we get everything out because we end up with a lot of substrate. And then from there, it's fairly simple. I just get them under the microscope and identify them using a dichotomous key. If for those who aren't familiar with a dichotomous key, it's just a series of steps. Each step will have 2 descriptions, and you keep running your specimen through, choosing which step describes your specimen, and eventually it gets to whatever taxonomic level you are trying to identify at. I identify largely to the level of genus. So the reason we use both eDNA and actual hand collection with physical specimens is that both methods have pros and cons. eDNA, it's great because it's fast, much faster than having to go through our specimens, 1 specimen at a time, identifying. And it also gets a much wider array of species from an area. But the drawback is that you can't really get abundance values. You just really get presence or absence. I believe some work is being done where they're working on methodologies to try and estimate abundance using eDNA, it's not really great for that, to my knowledge, at the moment. And for actual specimen collection and morphological identification, that's great because it can let you estimate abundance for each taxon. It also allows for other types of analysis if you need to do those, like you can look at different growth stages. If the organisms have morphological deformities, you can look at stomach contents, weight, that kind of thing. Uh, but the drawback there is it's much, much slower than using eDNA, and you find far fewer taxa. So for this project, we are using macroinvertebrates as bioindicators, so indicators of general stream health. And we're able to do that because each taxon is assigned something called a tolerance value from 0 to 10, where 0 are for taxa that are extremely sensitive to organic pollution. 10 is for taxa that are highly resilient. So if you're in an area, you collect a bunch of specimens, and you find a lot that have a very low tolerance value, that generally indicates little to no organic pollution in that habitat. But if you go to an area and you see mostly on the higher end of tolerance values, that indicates that there's likely some pollution source. And as we were analyzing the data, we use a number of different indices. One is called the Hilsenhoff Biotic Index. This uses those tolerance values I was just talking about and basically averages them for all the specimens in a sample and gives you a number from 0 to 10. And that tells you a little bit about the water quality. So here we have from the Hilsenhoff, the actual paper where this index was created and described. 0 to 3.5 indicates excellent water quality with no apparent organic pollution. And then on the other end, 8.51 to 10 is very poor, indicating that there's likely severe organic pollution present. And then another one, this is one of the more common ones, is the Index of Biotic Integrity, or IBI. This actually integrates a bunch of different indices, including the Hilsenhoff Biotic Index that I just mentioned. And this one is great because since it uses multiple different indices, it is a little less specific about what it tells you. Like if we were just using the Hilsenhoff index, that can tell us a bit about like pollution presence, but it doesn't give us much other information. Like it doesn't tell us much about the biodiversity or anything like that. But this integrates multiple different ones and gives you much more accurate information about just general stream health. And what we do for our analysis, we use values from the PADEP that gives us reference values to compare our values against. The DEP basically went to what they considered unimpacted streams or relatively unimpacted streams as like the ideal that we now compare our values against. So here I have highlighted the values they found. So if we're going to go to Hilsenhoff Biotic Index as an example, they have said 3.05 as like an indicator of ideal, relatively unimpacted stream. So if our values are higher than that, then that likely means that there's more pollution present. If it's lower, then it means that that stream has very little or no pollution. And then you standardize it, and you end up with a score between 0 and 100, with 100 being the most ideal. Here I have a graph of the taxa richness. This is data from 2021 and 2022. These are the four sites that I chose to talk about. Move this to the side. Here you can see total species richness. This is using eDNA. So we have some pretty large numbers at some sites. Like at this site, looks to be about 380 different species. But some sites are significantly lower, as low as looks to be about 40. So you can see very, very big ranges in how much diversity is present at each site. The four that we are looking at are somewhere in the middle, with the Upper East Branch being the lowest, Upper East Branch Calicoon. And then this is taxa richness data from last year. We collected from the same sites, same four sites, and this is using our hand-collected data, not eDNA, but you can see how different our taxon richness values are when we're using hand-collected specimens instead of eDNA, where our highest using the Serber sampler is a 46, so only 46 different taxa, compared to like this several hundred you get with eDNA. And just like in the previous one, the UEBC, that's the Upper East Branch Calicoon, has the lowest taxa richness, with the Serber and multi-hab being both about 20. And then here we have the EPT richness. Again, this is useful because these three groups of insects are especially sensitive. So if you don't see a lot of these, even if you have relatively high taxa richness, that can be an indication of poor stream health. And with the same 2025 data, uh, I'm comparing with the 2021 and 2022 data. This is the Hilsenhoff Biotic Index. So again, this is looking at the tolerance values. And here we can see that for most of the sites, we saw an increase in the HBI. That's an indicator that there might be more pollution present, except for a couple of sites, like here and with the Upper East Branch, actually, of Callicoon, we saw a decrease. So that could be an indicator of reduced pollution presence. And then using IBI, so multiple different indices, we see similar trends where unfortunately we saw a reduction in IBI scores for each site except the Upper East Branch Callicoon, which thankfully saw an increase. And this green bar is essentially indicator of like good quality. If it reaches this, that's good. If it's below that, it could be below average, but some are very, very close. And since we don't have the like error bars, I can't say statistical differences between years right now. Data analysis is still ongoing, and we still have a number of sites to collect data on from this spring. But for now, it looks like a trend for at least three of these four sites to see a reduction, although it's not always very significant. Another thing we found was the presence of four different invasive species. These were only detected with eDNA since all four of these species are terrestrial, so we wouldn't really expect to find them when we're collecting from the water. But these are the spongy moth, the viburnum leaf beetle, the red earthworm, and the hairy spider weevil. The three here, the two beetles and the moth, are all plant pests and could be important in that they are defoliating leaves, causing more sunlight to enter the streams, that kind of thing. And then the earthworm is generally less of a concern, I think, for most people, but they can contribute to greater erosion, especially along stream banks. And one of the takeaways here is in our eDNA data, we did not find the presence of Boyaria graphiana, the ocellated darner dragonfly, in 2025, even though it was detected, I believe, just in one site back in 2021. This is a vulnerable species, at least in New York, so it is something we want to keep an eye on. And even though we didn't detect it, that doesn't necessarily mean it's not present. Because it's a vulnerable species, it's going to be rarer than a lot of the other ones that we find. And so we just expect to find it less often. Uh, I'm hoping we'll be able to find it again with future monitoring, but it's just something we want to keep an eye on. And as I mentioned before, three of the four sites that we looked at here saw a reduction in the IBI values. Uh, until we start doing more statistical analysis, I can't really say what the cause would be, but we want to look into multiple different variables. Obviously, temperature is a big one, but weather events, so rain, cloudiness, that kind of thing. And of course, human development. I want to look into whether there has been significant development from some of these sites between 2021 and 2025, because that could also contribute. So as this research continues, we are going to continue doing statistical analysis, hopefully find out what factor is most significant. And as I mentioned before, we also have 23 more sites that we are going to be collecting macroinvertebrates from this spring. And that will give us more data to see if these trends are continuing. And because we are two different years, 2025 and 2026, hopefully, it will also help us determine how much influence weather patterns have, and if they are significant compared to just general temperature trends. We are also partnering with Dr. Stephen Ryer at Bloomsburg University. He is doing analysis of algae communities, so that's something we are looking forward to seeing. I'm curious whether the algae communities will follow the same trends as the macroinvertebrates, but that will just give us a little bit more data about stream health. And I would also just like to continue these monitoring efforts into the future. The more years of data we have, the more sure we can be of the actual trends. Because even if we see reductions or increases in certain indices, that doesn't mean that that's going to be a trend. It could just be one year there's a significant reduction for one reason or another. Again, weather events are pretty important. So it could just be one year had a lot more rain, and that can cause a lot of macroinvertebrate communities to get flushed further downstream. So it really requires multiple years of data to really be sure. And I would like to acknowledge uh, people at NIFWF, the National Fish and Wildlife Foundation, for their support. I'd like to thank Dr. Stephen Ryer at Bloomsburg, and of course, the Friends of the Upper Delaware River. And I will take questions if people have any.
Thank you for that, Duncan. Super interesting. I'm gonna stop the recording, and yeah, if anybody has any questions, please feel free to speak up or put them in the chat.